Hello, everybody. I want to welcome all on behalf of the RNA initiative at the Istituto Italiano di Tecnologia, and thank you, the organizer of the RNA collaboratory seminars and the RNA Society. Well, as usual, attendees are encouraged to ask questions through the Q&A panel of the Zoom interface. And this said, let me introduce the first speaker for today's session, Mariangela Morlando. Mariangela is an associate professor of molecular biology at the Sapienza University of Rome. After her PhD in biology at the University of Roma Tre in Rome, in 2006, she moved to the Oxford University, joining the laboratory of Nick Proudfoot, where she pioneered the study of microRNA biogenesis. In 2018, she established her own laboratory, focusing on the study of non-coding RNAs in neuromuscular disease, or ALS and DMD. Today, she will present her most recent data regarding a novel role for non-coding RNAs as crucial tethering molecules in regulating gene expression. Thank you, Mariangela.
Good afternoon, everybody. Can you see my screen?
Yes.
Okay, perfect. Thanks for this opportunity, and I would like to this afternoon to share with you our latest data regarding the role of non-coding RNAs in neurons. Just a brief introduction of the topic. Non-coding RNAs are transcripts that lack coding potential. They have different functions in cells. They can have structural functions such as ribosomal RNAs or tRNAs, but they can also have a regulatory function. And since always, I have to say, we are interested in the study of long non-coding RNAs in the linear and circular conformation. So regardless of their structure, linear or circular, the power of long non-coding RNAs in regulating gene expression resides in their ability to form a secondary structure and to interact with RNA and proteins, coding and non-coding RNAs. So they can work as a scaffold, guide, decoy, or riboregulators. This ability to gather together RNA and proteins allows them to build a ribonucleoparticles in which the single component can reach high concentration, leading to more efficient process or reactions. So because the cells is a crowded place, this is really important, mainly in cells which is highly polarized like neurons. And in this system, in this tissue, we wanted to analyze the role of circRNAs in creating such ribonucleoparticles. So circular RNAs are peculiar non-coding RNAs because they arise from encoding genes, actually through a peculiar splicing reaction, which is called back splicing, in which a downstream 3' splice site is joined to an upstream 3' splice site, leading to the formation of covalently closed RNA. This reaction can take place thanks to inverted repeats in the introns or through the intervention of RNA binding proteins. So this type of non-coding RNAs are really interesting because they are highly expressed in the nervous system. They are very stable, they are conserved, and they are very stable because they lack free ends. So they are good candidate to act as a building block for the construction of ribonucleoparticles. And in particular, some of them have been detected at the periphery of neurons. So they are really important also to exert function at neuronal periphery. So how they can work? Basically, they work as long non-coding RNAs, so they can interact with RNA and proteins. They can control the stability, the availability of such molecules. Few of them can be translated, and some of them present a nuclear localization and they are able to control transcription. So if we think of all these functions in terms of inside the neuronal system, we realize that all these functions can be exerted not only in the cell body, but also at the periphery. For example, controlling local translation. This means that the circular RNA have to find a way to travel along axons and neurites to reach the periphery. And at the synapses, they can also be secreted, so they may represent also molecule important for cell-to-cell communication. So our model system to study the role of circular RNA in neurons is based on in vitro derived neurons, motoneurons or glutamatergic neurons, derived from the differentiation of human or murine embryonic stem cells in the wild type condition or in a condition resembling amyotrophic lateral sclerosis. These yes, embryonic stem cells carry a mutation in a Fas protein, which is an RNA binding protein involved also in back splicing reaction. And when this protein is mutated from the nucleus where it normally localizes, it can delocalize in the cytoplasm. And upon stress in the cytoplasm, this protein can form aggregates that can, at the end, form this pathological inclusion that you can also see in post-mortem tissue from ALS patients. So we can recapitulate all these steps in vitro, so we can differentiate ES in motoneurons. We can have the delocalization of the protein and the aggregation upon stress we induced by adding arsenite to our culture. So in analyzing the expression of circular RNA in this system, we noticed that in Fas mutant condition, two circular RNAs conserved between human and mouse were deregulated. And today I will show you what we know about the function of these two circular RNA in neurons. The first one, called CIR31, is composed by 4 exons, while the second one is composed just by 1 exon. And I want to stress the fact that the circular RNA share most of the sequence with the linear counterpart. So you have just the back splicing junction, just this region to detect specifically the circular RNA molecules. So starting with the CIR31, we went to look what was its localization in motoneurons. And we used a particular fish technology called BaseCop, which makes use of 2 zeta probe designed across the back splicing junction. And only when these 2 zeta probe are very close to each other, you can use a third probe. And on this probe, you can build an oligo tree in which you can amplify the signal. Of course, this 2 zeta probe can also hybridize on the mRNA, but in this case, they are too far apart. So no signal comes from the linear transcript. So as you can see from this picture, you can easily detect circular RNA in neurons. And the interesting thing we observed was that the CIR31, the red spot, is localized in the cell body as well and at the cell periphery. So in these neurites, we cannot distinguish dendrites from axons, but still periphery of the neurons. And this localization is not affected by Fas mutation. So in wild type and mutant motoneuron, you still have CIR31 at the neuronal periphery. However, when you induce stress in these cells, so you induce the generation of the stress granules and Fas granules when Fas is mutated and it is in the cytoplasm, we observe a reduction of these neurites localization, both in the wild type and Fas mutant motoneurons. However, when you have Fas in the cytoplasm in the mutant condition, we observe that CIR31 was trapped in these Fas granules, suggesting that all the functions that this circular RNA may exert at the periphery are impaired. So circular RNAs, to exert this function, has to interact with RNA and protein. And we were able to observe the dynamic of the interaction between CIR31 and Fas granules also in vivo. thanks to this experiment that was performed by Erika Vitiello at the IIT in Genova, in which she labeled the circRNA with the aptamer that can be specifically recognized by this fluorophore, and you can follow. The red is the circRNA, and in green you have mutant Fas. You see that the connection between Fas and the circRNA is really tight. So in order to understand how this circRNA could function in motoneurons, we went to identify RNA interactors of the circRNA by using an RNA pull-down assay using biotinylated antisense oligonucleotides in presence of AMT, which is a chemical compound able to stabilize RNA-RNA interaction in a reversible manner. So we sequenced the RNA interacting with CIR31, we validated some of them, and we found mRNA encoding for translation factors, for endosomal and lysosomal proteins, for proteins involved in migration and neuronal differentiation, and transcription factors. Because we knew that in ALS condition, one of the process which is impaired is the local translation at the periphery, we focused our attention on the translation factor AIF3C, which shows the highest enrichment in the pull-down assay. And we went to check whether also this mRNA, as well as the CIR31, was trapped in Fas granules. And indeed, also AIF3C mRNA is trapped in Fas granules, but the interesting observation was that upon CIR31 depletion, this trapping is lost, suggesting that CIR31 is able to interact with this mRNA, maybe is responsible for its traveling along neurites and its localization at the neuronal periphery, where it can be locally translated and control, in turn, local translation of other targets. So this has a very interesting implication in ALS condition where local translation is impaired. So to just make first conclusions, I showed you that CIR31 is one of the circRNAs able to reach neuronal periphery. This neurites localization is affected by oxidative stress. And in ALS background, the combination of Fas mutation and the oxidative stress trapped CIR31 in Fas granules, and together with CIR31, all the targets. Regarding AIF3C, this, as I told you, has an important implication for the local translation, because if you trapped this mRNA in the cellular body, you lose its function at the neuronal periphery. About the other target, mainly for endosomal and lysosomal mRNA, we hypothesized that CIR31 may act as a tethering molecule between endosomes and this mRNA, encoding for protein that are important for the endosome function. And because it is known that some of these proteins are locally translated, it is possible that the circRNA is the molecule tethering these mRNAs on endosomes and allowing translation if it is needed. For CIRDLC1, we utilize the same fish approach we used for CIR31, so the base scope approach, but after the analysis of its expression in several neuronal districts in mice. Even though we firstly identified the circRNA in motoneurons, we realized that it reached its highest expression in striatum and in prefrontal cortex. And in particular, in the prefrontal cortex, the highest expression is in the deeper layer of this district, where the motor cortex relies. So, in order to understand the function of the circRNA in vivo, we raised a knockout mice, thanks to the collaboration with the INBL in Monterotondo, using the CRISPR-Cas9 technology. If you want to delete the expression of a circRNA, you have to remove the sequence important for circularization. So in this case, we removed the first intron, but we used an SDA DNA donor in order to restore the exon-exon junction, aiming to avoid the impairment of the mRNA production. So because of the expression, the circRNA in the corticostriatal districts, we went to test these animals in an open field assay. And we noticed that knockout mice showed a hyperactivity, disinhibition, and increase of stereotypies. All these behaviors correlates very well with an impairment in the corticostriatal circuitry. And this behavior is observed in several neurological disorders, such as hyperactivity. So in order to understand what was the molecular mechanism behind this behavior, we decided to focus on striatum because it is the district receiving many inputs for different districts in the central nervous system, including prefrontal cortex. So we analyzed the proteome of the knockout mice, of the striatum of the knockout mice, and in collaboration with Armilotti lab at IIT in Genova, and we observed that there were many proteins that were upregulated, and among them there was a group of protein belonging to the most enriched category in the geo analysis, and they are a subunit of the MDA receptors and AMPA receptors, together with this kinase, which is important for the recycling of this subunit. And this upregulation correlates very well with the electrophysiology we did in cultured neurons from these mice, showing that we have an increase of AMPA-mediated response. So all these proteins belong to glutamatergic synapses, which is exactly the synapses that forms between glutamatergic neurons from the cortex and medium spiny neurons in the striatum. So what about SIRDLC1? Which is the role of SIRDLC1 in this upregulation? So we went to look whether SIRDLC1 could affect the expression of the mRNA encoding for this upregulated protein via miRNA or via direct interaction with the mRNA encoding for this protein. So we got a list of miRNA expressed in the striatum from the literature, and in silico, we went to check whether there were some miRNA binding sites on SIRDLC1 on one side, and the miRNA binding site on the mRNA encoding for the upregulated protein. So by RNA pull-down assay, we were able to validate the interaction between SIRDLC1 and Miran-30B5P, and also by luciferase assay. And on the other side, we validated the interaction between Miran-30B and GRIN2A and GRIA1. For matter of time, I will show you results only for GRIA1, but we obtained exactly the same results from GRIN2A. On the other end, we also tested whether SIRDLC1 was also able to interact directly with the mRNA encoding for this upregulated protein. This is the in silico analysis, and by RNA pull down again, we were able to validate GRIN2A and GRIA1. So together with Miran-30B, SIRDLC1 is also able to bind the same mRNA. So focusing on GRIA1 and on the 3' UTR on GRIA1, we found that we identified 2 binding sites for the circRNA and 2 binding sites for the miRNA. So by using a luciferase assay, we tried to understand the role of each component of this regulatory circuitry, and we deleted CYP31 from cells, and we observed an upregulation of the luciferase activity, which parallels very well the behavior we see for GRIA1 protein in the knockout mice. GRIA1 was upregulated when the striatum was knocked out for SIRDLC1. On the other hand, when we block the activity of the miRNA, we still see upregulation, suggesting that also the miRNA is able to regulate GRIA1 expression. However, when we downregulate the expression of the circ and we block the miRNA at the same time, we observe a greater upregulation of the luciferase activity, suggesting that the 2 molecules can act in combination, in synergy, in regulating GRIA1 expression. So we also knew other things about GRIA1 regulation and min130B in neurons from the literature. We know that GRIA1 can be translated not only in the cell body, but also at cell periphery. And we also knew that min130B is one of the mRNA that can be detected at the periphery of neurons. So we went to look whether also in our system, in a neuron in culture obtained by differentiating in vivo mouse embryonic stem cells coming from wild type and knockout mice, we could detect min130B at the periphery. And indeed, by fish assay, we saw min130B not only in the cell body, but also the periphery in the wild type condition. But was really, really interesting to observe that in the knockout condition, so when you don't have circRNA in cells, we have a drop of the localization of this mRNA at the periphery. And in a very, very preliminary experiment, we reconstitute the expression of SIRDLC1 in the knockout background. And we not only recover the SIRDLC1 expression, but we also recover the min130B neurites localization. So SIRDLC1 somehow is able to localize min130B at the periphery, and in our model is also able to control the local translation of GRIA1. So if we want to build a picture of all this information, what our experiments suggest is that in a normal condition, in physiological condition, you have min130B in the cell body, but also at the periphery. And this localization is controlled by the presence of SIRDLC1. At the periphery, SIRDLC1 and min130B can bind the mRNA of GRIA1 and GRIN2A, controlling the translation. We still have to demonstrate that SIRDLC1 is also important for the loading of min130B on this mRNA. Anyway, in normal situation, you have a proper balance of AMPA and MDA receptors on the membrane surface, and you have normal synaptic activity. In the knockout condition, instead, you lose the min130B neurites localization or localization at the periphery anyway, and you don't have SIRDLC1. So the translation cannot be controlled anymore, and you have more AMPA and more NDA subunits on the surface of the membrane, leading to synaptic hyperactivity. So regarding this second circular RNA, I showed you that this circular RNA is highly expressed in the deeper layer of the prefrontal cortex and in the striatum. The knockout mice show hyperactivity and increase of stereotypic behavior, suggesting that in these mice, so you have an impairment of the corticostriatal circuitry. This might be due to the loss of regulation of GRIA1 and GRIN2A mediated by SIRDLC1 and min130B together. Also, this impairment in the cortical striatal circuitry has a very interesting implication in ALS, because one of the first process that it is affected in ALS patients is the cortical striatal circuitry. They show hyperactivation of the glutamatergic synapses even before the motoneuron symptoms occur. I would like to thank all the people that contribute to this work. First of all, Professor Irene Bozzoni and all the postdoc and researcher working in our lab, and our collaborators and findings, of course, and all of you for the attention.
Thank you very much, Mariangela. Really interesting. Are there any questions? Meanwhile, while we wait for people to ask any, I would like to ask you, Mariangela, do you have any data or any evidence regarding the protein interactome, or especially the SIRDLC1 circular RNA? Thank you.
Yeah, this is a good point. This is a part that we are developing now. I mean, we did pull down, followed by mass spectrometry. So we are in the way of validate some interactors. but we don't know any data that we validated, so we are sure about the interaction. But it is one part that we want to develop for sure. The things that we know that it interacts with ego, of course, because it interacts with the iRNA. So ego is there, but regarding the other protein, we're still working on that. It's not easy, yeah.
Yeah, very good. Thank you. Okay, if there are no questions for the moment, I would pass to the next speakers. Meanwhile, if anybody has any late curiosity, you can still write in the Q&A panel. So I move on to the next speaker, Francesco Di Palma. Francesco Di Palma is a computational biophysicist working as a senior postdoc in the group of Professor Andrea Cavalli at IIT in Genoa since 2019. His journey in the RNA world started in 2011 during his PhD at SISTA in Trieste under the supervision of Prof. Giovanni Bussi, where he focused on the simulation of the adenine riboswitch. Starting from this, he has scaled up the size of his atomistic simulations, and today he will present his recent findings regarding the eukaryotic ribosome. So thank you, Francesco.
Good afternoon, everybody. I'm sharing my presentation and begin. Okay, so first of all, I would like to thank the organizer for giving me this opportunity and all the RNA initiative at the IT for our interactions. And as Luca was saying, I will present you something about the ribosome stalling mechanism induced by the human XBP1U arresting peptide at atomistic level. So I would like to invite you at the beginning of this presentation to make kind of an experiment and close your eyes in order to imagine the inner part of a cell. How do you imagine it? And probably most of the computational biologists are able to imagine something like this, but not everybody. And this is kind of a crowd environment with all the water surrounding the different micro and micromolecules and small molecules like ions, metabolites, or even bigger molecules like tRNAs or proteins in this medium. Something that is missing in this picture is probably something that would have fit at this scale in my screen. That is the ribosome. This is because, as you probably know, the ribosome is composed of protein and ribosomal RNA for a total of approximately 380,000 atoms, and is composed of two subunits, the small subunit and the large subunit. It's the site of the protein synthesis, and here the mRNA is translated into the corresponding sequence of amino acids using the tRNAs. So in particular, in our research, we have focused on the XBP1U arresting mechanism, and this is a human Nash chain, a human protein that is synthesized by the ribosome, and while the synthesis is happening, there is something that the Nash chain induced on the ribosome itself for regulation purpose. Practically, while the Nash chain is exploring, is passing through the exit channel, the ribosomal exit channel, this Nash chain is able to stall the ribosome for regulation purpose. These purposes might be alternative splicing or membrane targeting of the protein itself. This is our system. So in this case, we are looking at a ribosome in shades with the tRNAs, eTRNAs and the ptRNAs in this solid structure, and as well as in magenta the mRNA, the small portion of the mRNA, and also the XBP1U arresting peptide in the exit channel. Zooming on the Nash chain on the right of this picture, we have highlighted the surrounding bases and amino acids of the walls of the exit tunnel, of the ribosomal exit tunnel, in order to let you understand where we are focusing our attention. So the XBP1U Nash chain is composed of 24 amino acids. In the exit tunnel, as I was saying, its structure is not linear inside the exit tunnel, with a turn close to the peptidyl transferase center. And as you can see in this pictorial representation of the ribosome, we have the ribosome that is in a post state, with only the E and the P tRNAs inside the structure, without any A tRNA. That is, in this case, due to the fact that the ribosome is stalled, is in a pose conformation, in a posing structure, there is no way for the tRNA to bind at its own spots in order to load the next tRNA and go on along the peptide synthesis. At the same time, there is no way for the eTRNA in this conformation of the ribosome to be released in order to go on. As you can see, the tunnel of the exit tunnel of the ribosome is mainly composed of the bases, the nucleotides of the 28S ribosomal RNA in the top part near the peptidyl transferase center. Of course, the peptidyl transferase tRNA on top. And whereas towards the exit, toward the mouth of the exit tunnel, we found two proteins surrounding the Nash chain, the UL22 and UL4. These are the main characters of our investigation, and our strategy in order to understand these the mechanism by which the arresting peptide is able to translate, let's say, this arresting mechanism to the ribosome. We have done equilibrium and non-equilibrium molecular dynamic simulation at atomistic level in the presence of different arresting peptide variants. Our idea, our strategy was to trying to let the ribosome try to escape from the arrest, and this is achieved by means of gently pulling out the peptide, the XBP1U peptide, by means of adiabatic bias molecular dynamics to study, of course, this stalling mechanism and its changes. What are adiabatic bias molecular dynamics? Mainly we can describe it as a sort of Paul-Ratched simulation in which we let the system evolve towards a target value in collective variable space. So we choose a variable for our simulation, and we use it. We set a target value for this collective variable, and we use an harmonic potential moving with the thermal fluctuation of the collective variable itself. In particular, this is a small video of a typical simulation with the ribosome in green and yellow, representing the ribosomal RNA and the proteins, and in red the Nash chain. In practice, we have that there is no bias potential if the system is moving towards the desired endpoint, whereas the simulation dumps the fluctuation if the system attempts at moving in the wrong direction. So if in this case we want the exit, the peptide exit from the translocon, from the channel of the ribosome, we set an ending point out of the ribosome itself, and we let the system evolve towards that ending point for our simulation. What we have done is to test by means of this technique not only the wild type arresting peptide, but also other 4 variants that we have chosen and modeled according to the mutation analysis in 2 papers of some collaborators. And what we have chosen, as you can see in this picture, are these 4 variants on the basis of 2 supposed completely different behavior from the wild type arresting peptide. In the representation of this mutation analysis, we can see which are the different variants at each position of the arresting peptide that would cause the production of more or less full-length protein. And so in this case, if we see a lower value of full-length proteins, this means that the arresting mechanism is stronger than the wild type. Whereas if we see something that is a higher value than the wild type, this means that the stalling mechanism is less pronounced. In this case, so we have chosen some single, double, and triple mutants, and we have done our different simulation. I will invite you to focus your attention on where these mutation are on the arresting peptide. If we divide the arresting peptide in 3 parts, with the C-terminal portion, the intermediate one, and the N-terminal, we can see that almost every mutation is in the intermediate part, where there is the turn inside the exit tunnel. And I will tell you something more in detail talking about the results in a few moments. At first, I would like to let you focus your attention on the protocol that we have used. We have done all-atom molecular dynamic simulation in explicit solvent using the Amber 14 force field defined with all the parameters for the RNA. And we have done at first a 1 microsecond long equilibrium simulation that was very stable. And you have to imagine that this system in this first simulation was 2.5 million atoms big. And after the modeling and the setup of the other of the mutants, we have simulated for 5 different systems, 20 replicas, 100 nanosecond long each, of adiabatic bias molecular dynamic simulation. And in this case, due to the fact that we need more space far from the ribosome itself, filled with the solvent, our box of simulation becomes even bigger with 3 million atoms in order to allow the full Nash chain to exit from the channel. These 5 variants were tested with different settings of the adiabatic molecular dynamic simulation, and in the end we obtained a total length of simulation of 10 microseconds. That is something that is probably never been tried on such a big molecules like the ribosome. So at first we have done destruction analysis on the stable microsecond long simulation, taking into account the different interaction that the arresting peptide is keeping or losing along this stable simulation. We have plotted here the number of contacts, intermolecular and intramolecular contacts, having an occurrence of lower or higher of 50 and 75% along the whole simulation. I would like to let you focus the attention on the green part, that is the one of the sequence of the arresting peptide, that is the one keeping more the mutation in our system in the second part, in the second set of simulation. But as you can see, there are a lot of inter and intramolecular contacts that are kept along the whole simulation. And for each residue of the arresting peptide, we have characterized the different contacts and interaction that are established with the wall of the exit tunnel of the ribosome, or internally, so in the between different amino acids of the arresting peptide itself. The second part of the simulation was the second set of the simulation. The aim of this second set of the simulation was, in some sense, to rank the 5 variants, the wild type and the other 4 variants of the arresting peptide in terms of kinetic and allow a comparison with the available experiments. So at first we have this is the report of how the collective variable progress along the different replicas for each system. As you can see, there is a particular behavior for one of the variants, that is the double mutant carrying a mutation at position 247, and another one at position 255, at variance with all the other 4 systems. In this case, we were not able to detach the different the amino acids of the arresting peptide from their initial position, and I will let you appreciate why, the reason behind this behavior in a second. Zooming on this plot, we can see that none of the replicas was able to progress along our desired protocol, our desired pathway. So with none of the arresting peptide with this carrying this double mutation to be able to exit from the arrested ribosome in order to release the stalling. This is an analysis in which we sum up the behavior of the different replicas of the different systems. As you can see, the wild type in the first row was able to detach from the peptide transferase centers in all the 20 replicas in a certain time, whereas, as I was saying, the double mutant, carrying the lysine mutation and position 247, was not able not to detach nor to fully extract from the exit tunnel in none of the replicas. So a real comparison with the experimental values I presented you before in the table at during a few slides ago was kind of possible in terms of the time, correlating the time that the methionine 260, that was the closer amino acid to the peptide transferase center to detach from its position, was possible. So in this case, we have that the lower time for the detachment corresponds to an experimental value of all the full-length protein that was effectively produced. So with a very little effect on the translation process. For example, instead, we have that in the presence of the triple mutant, instead, we have a almost the double of the time, or even more, the time was required for the methionine to detach from the peptidyl transferase center. And this is in agreement with a decreased experimental value for the full-length protein. So in this case, we have almost an order of magnitude of difference between the experimental values for the full-length protein that corresponds to always the double the time for the variant, the arresting peptide variant, to detach from the peptidyltransferase center. And what we can detail a bit more is where this delay in the detachment has been is occurring. And for example, if we take into account those simulations that were not able to detach from the peptidyltransferase center and to release the stalling, so in particular the double mutant, what you can see is that at a certain point there was no way for the amino acid 242 of the arresting peptide to move from its position. This is due to the fact that if you imagine that we are mutating a cysteine into a lysine, this is causing that the mutated lysine side chain, we carrying a positive charge on its tip, is facing all the negative charge of the phosphate of the backbone of the RNA, ribosomal RNA surrounding the exit tunnel. And in this way, facing all these negative charge, that lysine was not able to move from its position and to detach, and in order to let the ribosome release the stalling. What is happening in the case of the triple mutant, the green plot in this graph, is that at the exact position of the mutation that we had, there is a delay in the time that was required for that particular amino acid to detach from its original position. So in the case of the serine 247, here we have a delay, as well as at position 254, where a cysteine was mutated from a proline, we have another effect. On the contrary, in the presence of the mutation at position 255, where an alanine was placed in place of a serine, we have a reduced effect. And as well as at position 256, where a tryptophan was substituted by an alanine, we have that it was easier for the arresting peptide to be detached from its position. So meaning that the stalling is less strong in this mutant in comparison with the wild type sequence. We have detailed, again, the analysis in terms of inter and intramolecular interaction. If we take into account the double mutant that was not able to escape from the exit tunnel, you can see that almost all the interaction that were present at the beginning in the initial structure were not able to be disrupted along our simulation. And whereas in the presence of less stalling simulation, less stalling arresting peptide in the panel A, in the case of the tryptophan substituted by an alanine, we have that all at a certain point, very soon, all the interaction that were present between the arresting peptide and the wall and the basis, nucleobasis of the rRNA were lost at a very short time. So in conclusion, what we have done was to rank the wild type arresting peptide and the 4 variants with the single, double, and triple mutants in terms of detachment kinetics from the arrested stalled ribosome. The crucial aspect of the complex in the complex between the arresting peptide and the ribosome were captured thanks to our analysis. And in this way, we have revealed the interaction between the crucial interaction between the ribosome and the Nash chain that was inducing the stalling so stopping the translation, giving insight on the underlying arresting mechanism. And we have, of course, detailed the molecular explanation in terms of cause effect between point mutation of the arresting peptide and the arresting peptide potency and efficacy in terms of arrestings, of course. So this produced a very nice paper that was published in 2022 and in JCTC, as you can see. And so what's next? How we are going on this pathway? The idea that came up was to analyze in terms of putative, ligandable, or druggable pockets our simulation, because no one has such a big amount of data on a simulation of a full entire ribosome. So we extended our simulation, our plain simulation, the one at the beginning, to 10 microseconds, and now we are scanning the ribosome in terms of possible ligandable and druggable pockets in our simulation. This mainly for a reason, because if we were able to discriminate, for example, between in a particular spot that is on the ribosome between cancer or ribosomopathies in terms of possible binding pockets, this would mean that we were able to discriminate between mutated, so oncoribosomes, and wild-type ribosomes in order to develop a specific therapy for cancer and ribosomopathies. So at last, I would like to acknowledge the internal and external collaborators for this project, in particular, of course, Professor Andrea Cavalli and Sergio De Kerchi in our group at IT in Genova, as well as the collaborators in Stockholm University with Professor Gunnar Monainen, Sauro Succi at the ET at Rome, and Professor Michael Levitt at Stanford University. And of course, you for your attention, and the RNA initiative for the opportunity of giving this presentation. Thanks.
Thank you very much, Francesco. Waiting for questions. May I just ask a couple of them very briefly? So, first of all, is when dealing with millions of atoms at a time, what are the computational challenges linked to this? And then the second question is if this is feasible, have you ever considered exploring different concentrations of, for example, ions that are, for example, linked to altered cellular statuses in cancer, for example, to see how the perturbation of these might impact in the dynamics of translocation of this peptide? Thank you very much.
Well, the computational cost in this case was kind of important, and we were able to simulate this very big simulation box on a GPU machine that we have here in our institute. And you have to imagine that if for a normal protein, we are talking about, for normal protein simulation in explicit solvent, would make a simulation box of, I don't know, 100,000 atoms, or even bigger for a membrane protein. From channel through the membrane, we can reach, I don't know, 500,000 particles, so atoms. In this case, having 3 million atoms would itself was a challenge at the beginning to equilibrate the system. That was kind of difficult. But then, even if the simulation were not as fast, maybe as a normal simulation of a protein, maybe we were able to make kind of very long simulation in which we were able to find something interesting in terms, both in terms of the release, for example, of the arresting peptide that requires in our simulation, as I was saying, 100 nanoseconds, and but also longer equilibrium simulation up to 10 microseconds, as we have done. And so correlating somehow the times that was the biological time scale of what it was happening to our simulation. So we were able to effectively to simulate something relevant in terms of the biology of the system. Concerning the second question, I'm kind of in that direction, let's say, because more than changing the condition, for example, in the solvent, or in terms of counter ions or ions in our simulation, what we can, that is still feasible and can be done because of course if you set up the system in a different way with a different concentration of ions, you were able to probably to make different observation on the stability of the system, or the packing of the RNA, the ribosomal RNA, and everything else. Something that can be done, and it's in our ideas, let's say, is to for example to make our post-transcriptional modification on the ribosome itself, both on the protein, ribosomal proteins, or with the methylation of some important amino acid of the ribosomal proteins, or also to make some modification of the ribosomal RNA. So in terms of nucleobases that can be mutated, or as well as make mutations on the amino acids of the ribosomal RNA that can be, for example, peculiar of some particular cancer type, or in some ribomyosopathies. So that is the direction that we are trying to take, and the perspective ideas that I've shown you to look for putative druggable pockets is exactly that one. If we are able to make simulation of a mutant oncoribosome for a particular cancer type, and make the simulation of the wild type ribosome, and make a comparison in terms of accessibility of that pocket or targetability of that pocket, of course we will be able to hopefully to develop a specific target, a specific therapy, a specific and small molecule, for example, in terms of drug design or drug discovery, for that is able to discriminate between the wild type ribosome and the oncoribosome, for example.
Okay, thank you very much. Since no further questions, I would thank both the speakers. And for next webinar, which will exceptionally not be next week, but in 2 weeks. And in fact, next collaborative seminar is planned for the 3rd of July, and it will be hosted by the Helmholtz Institute for RNA-based infection research. So have a nice rest of the day, depending on your time zone. Thank you, everybody. Bye.
Bye. Bye.